Hi, experiment 4 is about preparation of triphenylmethanol. I am Ma.Alaiza M. Opina.
And I am Karen M. Estoya. So for the introduction, what is triphenylmethanol? It is a white crystalline solid that is insoluble in water and petroleum ether, but well soluble also in ethanol, diethyl ether, and benzene. In strong acidic solutions, it produces an intensely yellow color due to the formation of a stable triethyl carbocation. And it has many derivatives of triphenylmethanol, and this is where important dyes came from research laboratory. So for the structure, as you can see in the slide, uh, triphenylmethanol contains 3 phenyl rings and an alcohol group bound to essential tetrahedral carbon atoms. All the 3 carbon pH bonds are typical of sp3-sp2 carbon-carbon bonds with lengths of approximately 1.47 angstrom, while the carbon and oxygen bond length is approximately 1.42 angstrom. The presence of the 3 adjacent phenyl groups confers special properties manifested in the reactivity of the alcohol. For example, it reacts with acetyl chloride and not to give the ester but triphenylmethyl chloride. So for the properties of triphenylmethanol, its boiling point is about 360 degrees to 380 degrees. Its melting point is from 161 degrees to 164 degrees. Its physical state is that it is a crystalline and powder solid, and its color is off-white. And this compound is actually odorless for the odor. So for the uses, triphenylmethanol is an organic compound that is used as a reagent in the research laboratory. It acts as an intermediate in the production of commercially useful triaryl methane dyes. It is also used in the preparation of triphenylmethane. Triphenylmethane are used chiefly in copying papers, hectograph, and printing inks. It is also used as an anti-proliferative agent. An anti-proliferative agent is considered as antimetabolite, inhibits cell-cell pathways to limit T and B cell proliferation, and thereby reducing the cytotoxic response directed toward the cardiac allograft. So for the application, although it is not a major industrial importance, triphenylmethanol is a useful reagent in the research laboratory. Substituted derivatives of triphenylmethanol are intermediates in the production of the commercially useful triaryl methane dyes.
So in this experiment, we have 4 objectives. First is to prepare phenyl magnesium bromide. Next is to prepare benzophenone. The third one is to synthesize the triphenylmethanol. And lastly, to visualize and understand the mechanism of the reaction illustrated in this experiment. So shown in the slide are the reagents and apparatus needed to synthesize the triphenylmethanol.
So for the methodology, first part of the methodology is preparation of the Grignard reagent, the phenyl magnesium bromide. As you can see on the slide, the first part of the first methodology is to get a piece of magnesium ribbon out of 4.50 centimeter, about 4.50 centimeter long, and remove any oxide coating that may be presented or present by scraping both sides using spatula. And from the untarnished magnesium ribbon, weigh approximately 0.038 for its weight. For the second one, transfer the magnesium ribbon to the 25 ml round bottom flask using a micropipette and add 0.30 ml of bromobenzene. And also add 0.50 ml of diethyl ether to the flask. And if no reaction occurs, cautiously scrape using the spatula the surface of the metal in order to expose the unoxidized metal to the liquid. And when touching the flask, make sure to use a towel to protect your hands. When the reaction starts, you will see a slight cloudiness, then a brown color, basically brownish cloudy solution, which is visible in less than 5 minutes. Place the condenser in the round bottom flask with stir inside in a vertical position with cold water running. And mix 0.18 ml bromenzene and 1 ml of the ethyl ether in a test tube. For the sixth part, add the mixture, which is the bromobenzene and the ethyl ether, all the way through the condenser to the round bottom flask, 0.50 ml at a time using micropipette, and allow the reaction to subside before each addition. And the next one, insert the CaCl to sandwich with a piece of cotton at the top of the condenser when all the bromobenzene has been added to remove the moisture, and set the apparatus on a sand bath. Maintain at 60 degrees and allow the reaction mixture to reflux for 10 minutes. And afterwards, for the last part of the preparation of Grignard reagent, you need to place this setup in an ice water bath situated on a stir while waiting for the preparation of the benzophenone ether. So for the preparation of benzophenone ether, it is quite easy. You just need to obtain a 5 ml vial, and into it, transfer 0.238 grams benzophenone and 0.50 ml the ethyl ether. And for the next one, you need to warm this in a steam bath until it dissolves.
So for the synthesis of triphenylmethanol, first you must add the benzophenone that was synthesized from the previous method add the benzophenone solution to the stirred phenyl magnesium bromide 0.50 ml at a time. This time, the solution will turn bright red with the formation of chalky solid which gradually solidify. Next, remove the mixture from the ice water bath and swap it with sand bath when all the benzophenone solution has been added. So the third step is you must gently heat the mixture, allowing it to reflux for 10 minutes. Afterwards, replace the sand bath with an ice bath and allow the mixture to cool while swirling in vigorously, because the magnetic stirrer may not be enough to give an effective reaction. So next step is to neutralize the solution. So in order to neutralize the solution, add 0.50 ml at a time, 1.50 ml of 6 molar HCl. Note that the reaction is quite vigorous, so addition of hydrochloric acid should not be too hasty. Ultimately, the entire solid will dissolve and two distinct phases will form: the upper ether layer containing triphenylmethanol and the lower aqueous hydrochloric layer containing the inorganic acid. So the next step is to transfer the reaction mixture into a separatory funnel and draw off the aqueous layer. It is followed by to the ether layer, add small amount of anhydrous sodium sulfate stopper and shake it. Next is filter the solution using a quick fit funnel into a small Erlenmeyer flask. And lastly, heat the filtrate in a warm steam bath in order to evaporate the ether. Place also a stirring rod while heating. So in continuation, when boiling ceases, add 15 ml of hexane and stir the solution vigorously. White crystals of triphenylmethanol will separate this time. And next is collect the solid by vacuum filtrations, dry the crystals, and weigh. And lastly, Compute the percentage yield of the crude triphenylmethanol. So for the last method is the purification of the product. You must crystallize the crude product using hot isopropyl alcohol and followed by weighing the purified product and get the melting point and submit the product to your instructor. So let us now move on to the overview of mechanism of the reaction involved in producing triphenylmethanol. So the main goal of this experiment is to synthesize triphenylmethanol from bromobenzene and benzophenone. So to start the synthesis, a Grignard reagent must be prepared first via Grignard reaction, as you can see in scheme 1. A Grignard reaction reduces a carbonyl group into a hydroxyl group, and seeing that the hydroxyl group is in the form of a methanol attached to 3 benzene rings. After preparations of the Grignard reagent, the next reagent, as you can see in scheme 2, is a ketone with 2 benzene rings on either side of the carbonyl group, which is a benzophenone. More details will be discussed in the next slides. So the synthesis of a Grignard reagent was required for a Grignard reaction of this experiment to occur. So Grignard reaction is a nucleophilic addition reaction. A Grignard reagent, which usually is an alkyl magnesium halide, will function as a nucleophile and attack the electrophilic carbon in a carbonyl group, facilitating the formation of new carbon-carbon bond. Bromobenzene cannot undergo Grignard reaction on its own. It still needs magnesium to come in between bromine and carbanion. So to do this, addition of magnesium methyl to bromobenzene in diethyl ether is needed to begin the reaction. Diethyl ether is used because ether is an aprotic solvent, which means it does not have a proton attached to an electronegative atom, which also means it will not protonate the reactants. After which, phenyl magnesium bromide is formed. This is an important step before further proceeding with the preparation of triphenylmethanol. Ethers are a good solvent in Grignard reagent because of the ionic bond between magnesium and the halogen, which can easily be solvated by the oxygen of the ether, keeping the magnesium ion electrostatically stabilized. So let us now move on to the synthesis of triphenylmethanol. After phenyl magnesium bromide is formed, benzophenone can then be added. Again, benzophenone is an diethyl ether solvent. With the addition of benzophenone, the nucleophilic carbanion of the phenyl magnesium bromide now has a site of attack, as you can see in this slide. The very nucleophilic carbanion attacks the partially positive carbonyl carbon, pushing the pi electron towards the oxygen and therefore activating it. So at this point, the triphenylmethanolate is formed, as you can see in the next step in the mechanism. Triphenylmethanolate is just 3 benzene rings that are attached to the carbonyl atom with the oxygen of the carbonyl group, now only having 1 bond with the carbonyl atom and has an extra electron pair. Benzene is not a good living group, and since all other substituted attached to the carbonyl carbon are benzene ring, the carbonyl group cannot reform, so oxygen keeps it negative charge for the water work up. So water work up is simply water protonating the negatively charged oxygen, which is an alkoxide, to form the hydroxyl group. After the water work up, the synthesis of triphenylmethanol is complete, as you can see in the mechanism. The main point here is that the Grignard reagent attacked the electrophilic carbonyl carbon of the benzophenone and pushed electrons up to the oxygen. So the presence of acid allows for the now negatively charged oxygen to attack a hydrogen to form an alcohol. So in the synthesis of triphenylmethanol, shown in the slide is the sample data. So actual yield and theoretical yield is also needed to compute the percentage yield. Actual yield is the amount of product obtained from the lab data, while theoretical yield is the amount of product obtained from stoichiometry. The percent yield and purity equation is shown in the slide. So in conclusion, the reducing and R group delivering abilities of the Grignard reagent is tested by performing the Grignard reaction, which can be seen from the preparation of Grignard reagent, which is from bromobenzene, to the addition of the compound at which the R group is to be added, which in this experiment is the benzophenone to form the triphenylmethanol.
We met the objectives by virtually preparing and doing research for this experiment. And after doing extensive research, we finally visualized and understand the mechanism of the reactions that was explained in this whole experiment.
So that is all. Thanks for watching.